Hello, everyone, and welcome to today's webinar, Comprehensive Solutions for Your Treg Mouse and Human Research. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit miltenyibiotech.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. Our speaker will answer questions via email to the address you provided during registration. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Niloufar Monhasery, Global Product Manager for T-Cell Immunology, Miltenyi Biotec. Dr. Monhasery, you may now begin your presentation.
Hello everyone, my name is Niloufar Monhasery. I'm the product manager for immunology T-cells at Miltenyi Biotec, and in this webinar for today I'm going to explain you how to translate a Treg research workflow to clinical grade manufacturing. So let's start. The agenda for today. First of all, I would like to give you an introduction about Miltenyi Biotec, and then talking about Tregs, and then following the webinar by talking about Treg research workflow, and then Treg clinical grade workflow, and end up to the summary and final remarks. Miltenyi Biotec was founded at 1989 in Germany. We are more than 3,500 employees all over the world from more than 50 nationalities, and 25% of our employees are working at R&D, and we are reported in more than 46,000 scientific references. We have more than 17,000 products and presence in more than 100,000 cell therapy procedures, and also approximately 6,000 patients treated with cell products using our technologies, and we have direct sales in 23 countries, and we are transporting our products to more than 73 countries as well. Miltenyi Biotec is empowering discovery and advancing therapy. Our mission is to advance scientific research and medicine by providing solutions for curative cell and gene therapy, as well as biomedical research. We have a wide variety of product portfolio, and our products are from research to clinical applications. In Miltenyi Biotec, we are covering different topics such as immunology, cancer research, stem cell research, cardiovascular research, and neuroscience. And at the same time, in the clinical side, we have very great portfolio for graft engineering, immune therapy, and also regenerative medicine. Okay, let's start with some introduction about Treg, translating Treg research to clinical application. Regulatory T cells. Tregs are a very important subset of T cells, and they have three main functions. The first one is suppression of overwhelming immune responses, which keep the balance and homeostasis in the body. The second important functions is suppression of immune responses against self-antigens and non-self antigens, which protect against autoimmunity and mediate the tolerance in the body. And the third functionality of Tregs is suppression of anti-cancer immune responses, which support the tumor growth, and it's a functionality that needed to be considered, especially during the cancer treatment, as Tregs in this area are supporting the tumor growth, and their functionality needed to be controlled actually. And recognition of the regulatory T cells. As you might know, Tregs are not expressing any unique like surface markers. They can be recognized or phenotypes by combination of the surface markers such as CD4, CD25, lack of CD127, or CD45RA. and so on. And the very important intracellular markers, it's FOXP3, that expression of this transcription shows the functionality of this the Tregs. And also according to their origin of this Tregs, they can subdivide it in three groups: naive Tregs, peripheral Tregs, and in vitro Tregs. Tregs can be studied in different research and clinical fields and in different indications like transplantation, inflammation, allergy, cancer, GVHD, autoimmune disease, pregnancy, and so on. But why Tregs has really like key roles in the body? Because as you can see in this illustration, there is a balance between T regulatory cells and T effector cells in a situation that the body is in a healthy condition. But in some circumstances, this balance is disturbed, and Treg therapy is going to really like protect this balance and then retain this balance. So by expanding the Tregs and infusion to the patient, they try to keep this balance again, reestablish this balance again, let's say. So it's the reason that Treg therapy is an important part of adaptive immune therapy and T-cell therapy. And for all these research areas and clinical studies, you need to have good and optimized workflow in the research side and in the clinical side. And it's our mission to provide enough tools for you to achieve your goals. So let's go one step further and have a look at Treg research workflow at Miltenyi Biotec. Treg workflow consists of different steps: sample preparation, cell separation, expansion, and cell analysis. And as I've mentioned at the beginning of my talk, at Miltenyi, we are offering translational workflows, and Tregs is one of the topics that we are covering completely translational workflow. From research products, which I'm gonna show in orange color, that's our sort of products for human, mouse, and non-primate. And as you can see in this photo or slides, we cover all these steps of a workflow. And at the same time, we have some sort of GMP products that cover the clinical workflow. I'm going to talk about each steps in the following slides. Okay, let's start with research portfolio, and the first step is sample preparation. For getting a very good result out of your whole experiment, you need to optimize your sample preparation. The main resources of Tregs in mouse are spleen and lymph node, and also it can be isolated from tumor and other various tissue. And in human, we can isolate Tregs mainly from PBMCs, directly from blood, and also from tumor and other tissues. But in the sample preparation platform, we are focusing on the tumor and tissue material. And gentleMACST dissociator, big family, tissue dissociation kits, and all the solutions that you need to store your cells, remove your dead cells, and different type of tubes, and also strainers and filters that can help you to get a very high quality cell suspension with high viability are available in our sample preparation platform. I'm pretty sure that many of you know about gentleMACST, but I really want to emphasize on this great device and tell you that it is really, like I always say, that it is the heart of the sample preparation part, because it provides a standardized tissue dissociation, and you really save your samples, as the dissociation can be done in closed system, and it's one instrument for both cellular and molecular applications. And there are some optimized protocol for the most frequently used tissue, and at the same time customized programming for any type of tissue is possible in this device. And we offering you comprehensive kits for enzymatic treatments that is adapted and adjusted in this device as well. And we are offering elaborate strainers and filters to get single cell suspension and remove the debris and cell clumps, and all ended to very high quality with high viability of single cell suspension. Let's have a look at the application example that was done with one of our customers. They use gentleMACST dissociator and also gentleMACST C-tube, besides using 70 micrometer smart strainer, and then they try to dissociate the mouse dorsal skin, which is a hard tissue to dissociate, and then they identified FOXP3 positive regulatory T cells. As you can see here, the total number of the cells was compared with manual protocol and gentleMACST protocol. In gentleMACST protocol, you see that you have less variation. And in the next graph, you can see the reasonable and really like high difference between the CD4 positive FOXP3 positive Tregs out of gentleMACST in comparison to manual protocol, which shows the good efficiency of this device and combination of the products to dissociate the tissue. And also in this right hand side, you can see the FOXP3 cells which were analyzed with flow cytometry. That's the gentleMACST protocol showed a higher cell number of FOXP3 positive cells compared to the manual protocol. Okay, the next step, Treg isolation. You need to choose the best strategy to isolate your Tregs. At Miltenyi Biotec, we offer you wide variety of products. And I know that sometimes you say, which one is really fitting to our workflow, and how can I get high purity, high, let's say, recovery, and so on and so on. And some of you has very like specific needs in your workflow. But I can tell you, we have the max cell separation, magnetic separation technology that offers you different type of like kits, and we offer different and wide variety of the devices, from very manual devices to fully automated robotic integration devices. And beside all these separation kits, we are going to offer you Treg sorting with our unique MACSQuant Tyto Cell Sorters. I'm going to talk about each of these options step by step. Here you are, max cell separation. Select the best. We are offering you wide variety of products. For example, for positive selection, which is really ideal for rare cells and hard to process tissues. Or untouched selection with isolation kits, that is really like a good approach for pre-enrichment before fellow analysis or subsequent positive selection. Or MACSxpress solutions for really being fast and easy and convenient. And at the same time, you can scale cell separation according to your needs. You can benefit from completely manual approaches or benefit from MultiMACSx for fully automated system. In max cell separation, columns are a technological advantage. Max column amplifies the magnetic field by 10,000 times, and the cells get minimal labeling with nano-sized microbeads, and also cells-friendly hydrophilic coating and large space between steel spheres in column matrix are provided. And then you will enrich your cells without any specific activation. The cells has like free epitopes for downstream compatibility, and also there is no bit aggregation, as you can see in the picture as well. And this method can be easily translated to clinic, and it can be scalable and automatable according to your needs. And also this technology is used in clinical researchers for cell manufacturing in more than 55,000 patients, and it is approved by FDA. At Miltenyi Biotec, we are offering you a wide variety of products for isolating your Tregs. I know that sometimes it makes it really difficult for you to choose the best kit and a strategy for isolating your cells. So please stay with me. I'm going to explain you about all the strategy, then you can choose the best one for your research. First of all, I would like to start with MACSxpress Treg Isolation Kits, which enable you to isolate your Tregs directly from the whole blood. MACSxpress Treg Isolation Kit helps you to isolate your Tregs directly from blood. In this approach, your cells get labeled, and then depletion of non-CD4 positive cells can be done, and it followed by taking the supernatant, and then positive selection for CD25 can happen, and at the end the cells can be eluted in the final buffer. So in this approach, you will isolate your Tregs just in 30 minutes, which is quite fast. The current solution, other kits, takes longer than 90 minutes. And then you will get an excellent purity of the cells. As you can see here, 90% CD4, CD25 positive cells and over 60% FOXP3 cells. And also the recovery out of this kit is quite good. So Treg can be isolated from whole blood without phycol. And it's one of the options for the teams that time is a real matter for them. They want to get the best purity and recovery in a very short time. Let's continue with other kits that help you to isolate your Tregs from PBMC. Some of them are just for humans, some of them for human and mice. Let's have a look. In the next slide, you will see that how efficient each of them are working and how they fit in your workflow. All the Treg isolation kits based on two steps: depletion of unwanted cells and positive selection. And I put here CD4, CD25 positive Treg isolation kit as an example. In the first step, actually the depletion of unwanted non-CD4 positive cell can be done, and you will have untouched target cells. And in the next step, you will have the positive selection of CD25 positive cells. As you can see here, the untouched cells get labeled, and then they pass through to the magnet, and then the captured cells can be eluted to the final buffer. And you will have your final CD4, CD25 positive Treg cells. Okay, let's have a look at this busy slide. The first question is, what do you need? You really need to understand your application and why do you need your Tregs and what is your downstream application. When you can answer this question, then you can have a look at the different kits and then the different purity that you can get from each kit and also the recovery out of each kit. For example, if you are going for highest purity of the FOXP3 positive cells, like up to 96%, and isolate your naive Tregs, I would advise you to use CD4, CD25, CD127 theme kits. Or if you are really going for isolation of naive Tregs, but you want to expand them in long term in vitro, so I would suggest you to use CD4, CD49 negative Treg isolation kits. So, as I've mentioned, first of all, we need to understand why do you need your Tregs? What is your downstream application? And then we can help you to choose the best strategy and the best kit for your workflow. Okay, in this slide, I put some data for you that I know that you will like it. It shows the functionality of CD4, CD25 positive, CD127 dim negative Treg isolation kits. The fellow analysis shows that we can reach over 92% CD4, CD25 positive cells and also over 90% of CD4, CD25 positive, CD127 dim negative cells. And in the negative fraction, you will see that the ratio of the CD4, CD45 or a CD127 dim negative cells is not that much high. And it's, to be honest, it's one of my favorite isolation kits that helps you to really reach high purity with a very good recovery and also enables you to isolate your naive Tregs that you can expand for a long time in the culture as well. Tregs can be isolated via cell sorter as well. And here I would like to use this opportunity to talk about MACSQuant Tyto Cell Sorter. Many of you are already in the Treg field and know about this great device, as it is a translational device and you can use it in research and clinical sites. It's just one aspect of this device, but this device is quite gentle to the cells, then it helps you to reach high viability of the cells. It contains 3 lasers for up to 10 optical parameters, and also it's fully enclosed disposable cartridge for sample sterility and operator system, and you will get a very great viable cells that enables you to go for whatever downstream application that you need. MACSQuant Tyto cartridges are an important component of this system. They have 3 chambers: input chambers, positive selection chambers, and negative collection chambers. In the positive collection chambers, you will get your positive fraction, and in negative collection chamber, you will have the negative, let's say, sort of cells. And then in the negative fraction, you can even use them for the second round of selection. And as you can see, the microchip is located here on the bottom of the cartridge, and you can see the size of this microchip, which is quite small. And also the maximum volumes for the input is 10 milliliter. Positive collection chamber can contain 2 milliliters, and then negative collection chambers 10 milliliter. And as you can see here, this sorting, this valve is it's the valve that just direct the negative fraction to the negative chamber and positive fraction to the positive chamber. And here are located these 3 lasers that help the cells to be sorted accordingly. In this slide, you can see an example of sorting of memory Tregs. To get the memory Tregs, we stain the cells with CD4, CD25, CD127, CD45RO, and CD45RA fluorescence colors. And then we sorted the cells, and the target cells you can see for CD4 positive, CD25 positive cells will reach up to, over to 95%. And like naive Tregs will reach like 97%, and the memory Tregs fraction is really great number, like over 98% purity. And it shows the functionality of this cell sorter for sorting even like the very, very rare cells with the highest purity that you can get. Treg expansion. I know that this step can be very challenging, but with choosing the best component for your Treg culturing, you can optimize this step. MACScell culture media offering you TextMACS medium, which is a special medium for expansion of T cells and also Tregs. They are free of animal-derived component. They are offering optimal cell growth, reliable activation, and also are available with or without phenolreds, and they are available up to GMP qualities, especially attractive for the teams that has like translational plan to translate their research studies to their clinical studies. The second component is the choosing the good cytokines. We offer you max cytokines in premium grade, which has improved sequence that offer you better reconstitution and increased stability and activity in the culture. And also they are available for human and mouse cell stimulation. They give you a lot of specific biological activity, and also they are available up to GMP quality. The next component is expansion kits, which help you to simulate your Tregs to get the best, let's say, expansion. We have these expansion kits for human and mouse, and also these expansion kits are available to GMP quality. In this slide, I would like to show you with some data the efficacy of these components all together. Okay, we decided to isolate the Tregs with different isolation kits. And we did that, and then we cultured the cells in TextMACS medium in the presence of IL-2 premium grade rapamycin and the simulation kits or expansion Treg kits. And I just decided to show you the expansion rate of the Tregs out of MACSxpress Treg Isolation Kits. After 14 days, we reached 22 times expansion. And after 21 days, we reached 40 times expansion rate, which is a great number. But it is not the end of the story. Your cells needed to be really functional cells. You can test the functionality of your cells with some like functional assays, but at the same time, it's really important to check for the expression of FOXP3 to get the most inhibitory cells in your final yield. And as you can see here, over 67% of the expanded cells are positive for FOXP3 cells, which is a great results. Okay, at this point, I want to stop with talking about Treg research workflow and want to start with the Treg clinical grade workflow. And at the end of this step, I'm going to talk about cell analysis as well, because the cell analysis or Treg analysis platform for Treg research workflow and Treg clinical workflow is more or less similar. Then I would talk about it at the end of the Treg clinical grade workflow. I hope that you can remember this slide. As I've mentioned, we are a translational company and we have solutions for the research studies and clinical studies. We already covered the research side, but as I've mentioned, I'm going to talk about cell analysis at the end. And then I want to start with the clinical grade Treg manufacturing and our solutions for you. So let's start. There are three main steps for clinical Treg manufacturing: Treg isolation, Treg expansion, and Treg analysis. And as you can see here, I've sorted all the needed material that you might need for producing clinical grade Tregs. I'm going to talk about each step in the following slides. For clinical grade Treg manufacturing, we are offering you two workflows, but these two workflows are different just in Treg isolation step. In the workflow one, you can see that CliniMACS Plus is the main device that you can deplete CD8 and CD19 cells and also enrich the CD25 cells. And this step can be followed by cell sorting with a MACSQuant Tyto Cell Sorter for getting a higher purity even. But in workflow 2, we have CliniMACS Prodigy LP25 process, which consists of 3 steps: enrichment of CD25 cells, labeling the Tregs with fluorochromes, and elute the Tregs in Tyto cell sorter buffers. And then the cells pass through to the MACSQuant Tyto Cell Sorter for reaching the very high purity cells. Okay, I would like to give you more info about this part, as I know this step is really like challenging for lots of you, and you know, and you're very like interested to know more about it. Workflow 1, which is Treg isolation with CliniMACS Plus. It's specifically a proper approach for producing cells for example, GVHD prevention and treatments, and suitable for the application that we need to reach intermediate to high purity. And you can reach like less than 96% Tregs, and these manufacturing times, or better to say the isolation steps, takes 7 hours, and you can expand your cells according to your application from 12 to 36 days. But there are different like workflow, different approaches that you need to see which approach and which method for expansion of your cells is fitting best to your application. And final cell numbers, depending on the donor and the expansion time, absolutely it differs, but we expect to get like 50 to 1,000 times expansion. As I've already mentioned, in CliniMACS Plus, you can deplete your cells, CD8 and CD19 cells, and also enrich CD25 cells. And we have all the reagents in GMP grade. And afterwards, it can be followed by cell sorting to get a higher purity. But in these graphs, you can see that you can reach like over 92% purity or target cells, CD25 positive, CD127 dim. Okay, CliniMACS Plus device offers you different aspects like convenient isolation process, very high recovery, high cell number yield, and maintenance of Treg phenotype, maintenance of Treg function, and also polyclonal isolation. And we have a reference list as well that you can use and see which teams are using this system and how efficient it's working. But what is interesting at this point is that there are two main questions in the field right now. That's how to isolate different Treg subsets or antigen-specific Tregs, and how to reach the ultra purity Tregs, because of some application right now in the market that many of you need to really reach ultra pure Treg cells and at the same time isolate your antigen-specific Tregs or very rare Treg subsets. So, what is our solution for this step? Okay, the challenges in the market. Naive Tregs is a small cell population, and I know that many of you need to isolate the naive Tregs for downstream applications. Ultra pure Tregs is another challenge that there are lots of like opportunity for sorting the cells, but they are quite stressful for the cells. And as I've mentioned at the beginning of my talk, Tregs are quite fragile cells. So the quality of the cells after the cell sorting is not really the proper one. And also GMP grade flow sorting is needed, and it is missed actually right now. But we as a tool provider offering you two devices, CliniMACS Prodigy with LP25 pre-enrichment process and MACSQuant Tyto Cell Sorter, which gives you this opportunity to isolate different Treg subsets and reach ultra pure Treg populations. So let's have a look on the following slides and see how these devices can help you to reach your goals. CliniMACS Prodigy LP25 process enables you to enrich your CD25 cells and at the same time help you to label your Tregs with the proper fluorochromes and then elute your Tregs in a special buffers that can be transported to the MACSQuant Tyto Cell Sorter, and then you can sort your cells to isolate the subpopulation that you want to reach and also reaching ultra pure Tregs. And this application is mainly used for autoimmune diseases, for producing CAR Tregs. In organ transplantation, you need to really isolate a specific antigen-specific Tregs and reaching ultra pure Tregs, GVHD. And in summary, it's really suitable for application which need ultra pure Treg subset. And in this approach, you can reach over 98% purity. And the isolation part takes like about 10 hours, and expansion, as well as I've mentioned in the previous case as well, it depends really to your protocols and which approach you want to use and what is your downstream application and how much you need to expand your cells, and it can be vary from 12 to 36 days. And the final cell numbers, it's again dependent on the donor and the expansion time, but it's normally needed to really reach 50 times to 1,000 times expansion. As far as Tyto Cell Sorter is one of the very well-known device for sorting of Tregs in Treg therapy field, I would like to talk about it more again and emphasize on this device and the beneficial points and the advantages of the device that you can really use in your workflow. You can use TytoCell Cell Sorter with GMP grade cartridges to reach really like a trial and close system. No carryover or cleaning is needed. It's plug and play, easily scalable, no droplets or detectable aerosols, and sample is never lost. And all can be done in the cartridges. Okay, I would like to talk about the capacity of the cartridges, which I know is really important in the clinical applications as you have more samples and you need to save your times and also be flexible with different volume of the cells. Here I would like to show you that we have one standard cartridge and the other model of the cartridge is high speed cartridge, and it's an amazing, let's say, cartridge that helps you to save your time, especially when you have high volume of the sample to sort. And look, liquid flow is in a standard cartridge for 4 milliliter in an hour, but in high speed cartridge you can analyze 8 milliliter per hour is the samples, and inputs for both of them are 10 milliliter, and maximum cell concentration in a standard cartridge is 50 million per ml, and also in highest speed cartridge. But what is different is maximum cell flow rate, that in a standard cartridges you can analyze 55,000 cells per second, whereas in highest speed cartridges you can analyze 110,000 cells per second. And as you know, and you can see here, you can really like save times in this way. For example, in each sort for in standard cartridge takes like 2.5 hours, whereas in highest speed cartridge 1.25 hour per sorting it might take time. And regarding to the Treg sorting, as they are rare cells and you need to start with really like high volume of the samples, I can imagine that high speed cartridge fits perfectly into your workflow. Another aspects of MACSQuant Tyto Cell Sorter, which makes it really fitting into clinical setting, is that sorting happen exclusively in the fully closed and sterile cartridges, and there is no fluids inside the instruments itself. And also currently it is approved for 10 different clinical trials, phase 1 and 2, and over 200 patient samples sorted with Tyto Cell Sorter. And also all the consumable are available in GMP grade, max GMP grade, and all the extensive documentation are available for all that touches the cells. And we provide you max GMP Tyto cartridges, max GMP Tyto running buffers, and the last but not the least that I know might be very attractive for you is max GMP fluorescence antibody, that we have a very optimized and complete panel of antibodies for sorting Tregs. In this slide, I would like to share some data with you to show you the efficacy of MACSQuant Tyto Cell Sorter in sorting the naive Tregs. On the left-hand side, you can see the analysis of the cells, which are post-Prodigy LP25 process, pre-Tyto cell sorting, that in this graph, it shows that we reached 38% pre-enrichment of the Tregs. It was followed by debulking sorting on MACSQuant Tyto Cell Sorter, and we reach 83% purity. But as far as we need higher purity or go for ultra pure Tregs, we started another sort of purity sorting. And the final yield shows you that we reach 98% pure naive Tregs, which you can see are positive CD25 and negative for CD127s. Treg expansion. Again, in the clinical grade Treg expansion as well, there are lots of challenges. Miltenyi Biotec is offering you a customized solution to automate your workflow. You need to ask our customized application CAP team to design a proper process for you. Otherwise, we have some sort of products that helps you to expand your cells. But if you want to benefit from automation in a closed system, you can contact us and ask for customized application, and our application people are ready to help you. Okay, Treg expansion as well in a clinical grade needed some component that I would like to talk about them in the following slides. I have already talked about TextMax medium in the research side, but as I've mentioned, we have the GMP medium grade as well, which contains pharmaceutical grade human serum albumin, and it has optimized formulation with or without phenol, and it has QC functionality tests in every batch, and it's filled in flexible bags as well, or you can ask for customized format and according to your request, our team proper the proper format for you. But don't forget that it is really specialized for optimal cultivation of human T cells and Tregs. Okay, you need definitely 3 more components to reach very good expansion of your Tregs. Max GMP expect Tregs bits. It's an stimulatory bits that help you to expand your Tregs, and it maintain FOXP3 expression in your cells. Max GMP rapamycin that you need to use to inhibit the expansion of the conventional T cells, and also to enhance the proportion of expanded Tregs, FOXP3 cells in culture. And also max GMP IL-2, that you can see the functionality of this max GMP grade cytokines compared to the other commercial products as well. In these slides, I would like to share you some application data. You can see that after sorting of your cells and Tyto Cell Sorter in this experiment, we reached 92.7% FOXP3 positive cells and CD25 positive cells. 14 days culturing in TexMACS GMP medium, MACS GMP Expect Tregs Kit in the presence of rapamycin and GMP IL-2, you reach 98.3 positive positive cells for FOXP3 and CD25. But don't forget that Treg expansion is really donor dependent and depending to the fact that you are using the healthy or patient samples, depending on your manual protocols, and depending on the duration of the expansion. And all the components that are present in the culture play a key role on the expansion rate of your Tregs. For Treg analysis, you can analyze your Tregs functionally or phenotypically. And there are some key markers that you need to analyze them during the phenotypical analysis. And for functional analysis, we have a specific Treg suppressor inspector kit. To analyze your cells properly, you need to use a good, let's say, fellow cytometry device. We offer you MACSQuant analyzer family with different definitions and models. I don't want to really talk about all of them in the details, but just to informing you that we are offering you a big family of the MACSQuant analyzer with different specification. For phenotypical analysis of Tregs, we have some Treg detection kits, which are optimized antibody kits with titrated or let's say pre-titrated antibodies developed as a simple one reagent solution. And at the same time, you can benefit from reaffinity recombinant antibody plus our VioDye antibody conjugation, which offers you high purity and lot-to-lot consistency for greater reproducibility, and you can eliminate tedious and costly Fc receptor blocking steps because they have one universal isotope which control for the convenience and cost saving. And here I would like to tell you that we can help you to analyze your cells phenotypically with very optimized and flexible panel of antibodies. You just need to contact us and say your aim, your goal, your wish panel, and we help you to optimize your panel. Here I can show you like Treg surface marker detection using an 8 color panels. We can help you to even do the correct gating and designing the proper panel for your specific needs, and also staining the intracellular markers and surface markers, and then the cells from different resource cells. And also you can analyze the functionality of your Tregs. And Treg suppression inspector is a polyclonal stimulus that are MACSI beads coupled to alpha CD3, CD28, and CD2. These MACSI beads coupled with this alpha CD3, CD28, and CD2, they stimulate your responder T cells in an optimized way that they can proliferate. As you can see here in this illustration, the cells, the responder cells, got the simulation and they expanded. But in this part, they got the simulation, but as far as the T responder cells are co-culturing with Tregs and the Tregs are functional, they didn't let the T responder cells to be proliferated. And in this way, you can analyze the functionality of your cells. I really wish for longer time to show you more application examples and cover all the aspects of the workflow and all the solutions that we have for each steps, but please reach out to us to get more information. At this time, I have to really go to the summary part and just finish my talk. If you need more information, on our webpage, you can find our clinical and research brochures. We have different application notes, references list. We have like different type of scientific posters, Max and more, some customer reports, and you can have a look at the application page for the research regulatory T cells, clinical application of regulatory T cells, and getting lots of information about different devices that I was talking during my talk. And don't hesitate to contact us, the local team and global team directly to get more information in each step of your workflow. And with this disclaimer, I would like to finish my talk and say that I'm so thankful for your attention, your participation.
Thank you again, Dr. Monhasery, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone, and goodbye.